Assalamu alaikum dear student, let me discuss with you the most important topic of bioenergetic, that is glycolysis and its pathway in detail. Actually, glycolysis is the first step of cellular respiration in which there is breakdown of glucose into 2-pyruvate molecule. It is that step of cellular respiration in which there is the breakdown of glucose occurs into a 3-carbon compound called pyruvate. So the breakdown of glucose into a 3-carbon compound called pyruvate is called glycolysis. This glycolysis occurs in the cytoplasm of the cell, and this glycolysis is also called Embden-Meyerhof pathway because the scientist Embden-Meyerhof discovered it, so it is also named as Embden-Meyerhof pathway. This glycolysis, remember, this is the MCQ's point. Glycolysis is the anaerobic step of cellular respiration. It means that glycolysis does not need any oxygen to occur. So glycolysis can occur without oxygen, so therefore it is also called the anaerobic respiration. Glycolysis produces the net product in glycolysis is 2 ATP, 2 NADH, 2 and 2 pyruvate. The glycolysis occurs in two steps. The glycolysis occurs in two steps. One step is called preparatory phase or energy investment phase because in this phase energy will be used, and the other phase is called oxidative phase or energy yielding phase because in this phase the energy will be produced. Now glycolysis, you can see in this flowchart diagram. Here I will explain the whole process through the whole pathway of glycolysis. Here you can see glycoglucose is converted into a 3-carbon compound called pyruvate, and this whole process is called glycolysis. Glyco means glucose and lysis means breakdown, so the breakdown of glucose into pyruvate. This occurs now in the first step, glucose is converted into glucose 6-phosphate by an enzyme called hexokinase. In this reaction, ATP molecule is converted into ADP. Here one phosphate group is given to the glucose molecule, and that phosphate molecule attached to the carbon number 6, therefore glucose becomes glucose 6-phosphate because on the 6th carbon there is the attachment of phosphate group which comes from the ATP molecule, so the glucose is converted into glucose 6-phosphate. This is the first step. In the second step, only isomerization occurs. Now glucose 6-phosphate is converted into fructose 6-phosphate. There is only the conversion of this aldehyde functional group into a ketone functional group, therefore glucose is converted into fructose. Now on the carbon number 6 there is phosphate group, therefore glucose 6-phosphate now is converted into fructose 6-phosphate by enzyme called isomerase enzyme because this step involves the isomerization process in which the aldehyde group is converted into ketone group. Now in the next step, this fructose 6-phosphate is converted into fructose 1,6-bisphosphate. This reaction is catalyzed by kinase enzyme, and again here one ATP molecule is converted into ADP and another phosphate group is added and the carbon number 1. So here we can say fructose 6-phosphate is converted into fructose 1,6-bisphosphate because there is the attachment of phosphate group on the carbon number 1 and as well as on the carbon number 6, therefore it is called fructose 1,6-bisphosphate. Now the fructose 1,6-bisphosphate is broken down into two pieces. One part is called dihydroxyacetone phosphate and the other part is called glyceraldehyde 3-phosphate. Now this fructose 1,6-bisphosphate is converted into two molecules of 3-carbon compound which contain one molecule of them contain ketone group, so it is called the dihydroxyacetone phosphate, and the other molecule contain the aldehyde functional group, it is called glyceraldehyde 3-phosphate, and this reaction is characterized by aldolase enzyme. And you must know that dihydroxyacetone can be converted into glyceraldehyde 3-phosphate, and these reactions are interconvertible into each other by enzyme isomerase. Now from glucose to glyceraldehyde 3-phosphate, in this step there is the use of ATP molecule in the step 1 and in the step 3. Therefore from glucose to glyceraldehyde, this step is called oxidatory. This step is called energy investment phase or preparatory phase. Now this glyceraldehyde is converted into 1,3-bisphosphoglycerate. Here the NAD molecule is converted into NADH, and this reaction is catalyzed by an enzyme called dehydrogenase enzyme. Now the glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. Now the 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. The phosphate which is attached to carbon number 1 is released, which convert the ADP molecule into ATP, and this reaction is catalyzed by phosphoglycerate kinase enzyme, and here is the formation of ATP molecule. Now the 3-phosphoglycerate is converted into phosphoglycerate. Only there is the change in the position of phosphate group. Now the phosphate group from carbon number 1 is shifted to carbon number 2. Now the phosphate group from carbon number 6, carbon number 3 is shifted to carbon number 2. Now it is called, now it became from 2-phosphoglycerate to 2-phosphoglycerate, and this reaction is catalyzed by enzyme called mutase. Now the 2-phosphoglycerate is converted into phosphoenol pyruvate, and here is the removal of water, that is dehydration of 2-phosphoglycerate occurs, and which results into the formation of phosphoenol pyruvate, and this reaction is catalyzed by enzyme called enolase. Now the phosphoenol pyruvate is converted into pyruvate, and again there is the release of phosphate group, and this phosphate convert the ADP molecule into ATP, and this is the last step of glycolysis, which is characterized by pyruvate kinase. Now in this step, from 1,3-bis-phosphoglycerate to pyruvate, there is the formation of ATP molecule. So this phase is called oxidative phase or energy yielding phase. Now we can say that there are 3 irreversible steps in the glycolysis. You can say that glycolysis involves 3 irreversible steps. Which one? This, the first step, in which glucose is converted into glucose 6-phosphate, characterized by exokinase. This step is called the irreversible step of glycolysis. And again this third step, in which fructose is converted into fructose 1,6-phosphate, and which is characterized by phosphofructokinase. This is also the irreversible step. And the last step, in which phosphoenol pyruvate is converted into pyruvate by pyruvate kinase. These, the first step, third step and last step, these 3 steps are considered as the irreversible step of glycolysis, in which which cannot be reversed. Now there are 3 irreversible steps in glycolysis. Which one? Step 1 characterized by exokinase, step 3 characterized by phosphofructokinase, and last step catalyzed by pyruvate kinase. Now what is the clinical importance of glycolysis? The deficiency of enzyme pyruvate kinase, which was involved in the last step of pyruvate, which was involved in the last step of glycolysis, that was when phosphoenol pyruvate was converted into pyruvate, and this step was characterized by pyruvate kinase. If the deficiency of pyruvate kinase occurs, if there is no pyruvate kinase, then the phosphoenol pyruvate cannot be converted into pyruvate. So when the phosphoenol pyruvate is not converted into pyruvate, then there will be no formation of any ATP, and there will be the depletion of energy, there will be decrease in ATP, and this decrease in ATP causes an anemia called hemolytic anemia. This is the most important clinical importance of glycolysis. When the last step of glycolysis does not occur, and it causes a disorder called hemolytic anemia. Here I complete the discussion of glycolysis. Let me revise it. What was glycolysis? Glycolysis was actually the breakdown of glucose into pyruvate molecule through a series of reactions in which glucose was converted into 2-pyruvate molecule, and this step occurs in the cytoplasm of the cell, and it yields to it